Okay, I'd like to introduce our next presenter, Dr. Vicki Wheelock from the University of California, Davis.
Excellent. Okay, all right. Good morning, and thank you. I want to thank the organizers for inviting me to come and speak to you about our program at UC Davis to develop a cellular therapy trial for Huntington's disease. So Huntington's disease is a terrible, fatal disease. It's hereditary. It causes thinking, movement, and behavioral problems. It's a relatively rare disease. Perhaps 30,000 people in the United States have Huntington's disease, with about 2,000 diagnosed every year. But because it's hereditary, there's a pool of about 150,000 family members who are at 50% risk of getting this disease. It has a worldwide occurrence in all populations. Death is inevitable, and the estimated annual costs in the United States of this disease are on the order of $2.5 billion. Huntington's disease is attained through genetics. So every child of a parent who has Huntington's disease has a 50-50 chance of inheriting the gene for the disease. And tragically, Huntington's disease presents much earlier than other neurodegenerative diseases like Parkinson's and Alzheimer's, right during people's midlife, right when they're attaining most of their goals in terms of work and profession, and also when they're doing important work in their personal lives, having families, raising children. So it's a double whammy. The gene for Huntington's disease was found back in 1993 on the short arm of chromosome number 4. And I think many of us thought that there would be a cure right around the corner after the gene was found. But obviously it's been a very, very difficult process. So the Huntington's disease mutation is to have an amplification of the CAG repeat length in the Huntington's disease gene, in the Huntington gene. And people who have the mutation have greater than 38 CAG repeats in a row, and normal is less than 31, and commonly a normal allele has 17 or 20 some CAG repeats. The CAG repeat length ends up causing the protein to have a polyglutamine chain, and it's felt that that polyglutamine chain is important in the pathophysiology of the disease-causing symptoms. If we look inside the brain, I should start off by saying that this mutation is present in every cell of the body, but there's selective cell death within the brain. And if we look inside the brain at the medium spiny neurons that are deep within the brain in an area called the striatum, they're getting sick and dying out in huge numbers. If we look at longitudinal MRI scans over time, we can see this progressive death of cells that's going on primarily within the striatum. But if we keep looking in this longitudinal study at patients with early, mid, and late-stage Huntington's, there's this tremendous degeneration in the cortex and the white matter, and patients die mute and akinetic and rigid and bedbound. Treatments for Huntington's disease are strictly palliative. I'm an expert in the palliative treatment of Huntington's disease. That's what I've been doing for years and years. And it's heartbreaking work. The only thing that keeps us going is the research, and the community of families with Huntington's has participated in research studies for many years. They've really come together. They've been strong advocates for stem cell research. So we have one drug approved by the FDA to treat the chorea, and everything else we do is palliative. We're very fortunate. In 2007, UC Davis recruited Dr. Jan Nolte to come to start our Institute for Regenerative Cures. Dr. Nolte, like many stem cell researchers, works on a number of different diseases, and she hadn't worked on Huntington's disease before she came to UC Davis. But we were very fortunate that she met with our clinical personnel, she met with the family, she became inspired, and she's been working very, very hard to develop treatments for this terrible disease. And she had an extraordinary idea about how to treat Huntington's. And her idea was to use the adult stem cells, mesenchymal stem cells, which are uniquely suited to being a treatment vehicle in this disease. So the advantages of these cells would be the long safety record. They're not pluripotent. They're adult stem cells harvested from bone marrow. They have special properties of homing in on sick and dying cells. They are immunologically privileged. They shelter themselves from the host immune system and can be transplanted without tissue matching. They regulate inflammation, they secrete factors that help to support neuronal survival and connections, but most importantly, they can be easily and safely engineered to transfer molecules and proteins to target cells. So they're an ideal delivery system. So back to the Huntington's disease mutation. After all these years, we still don't know exactly why the Huntington's mutation causes these cells to die out. We have lots and lots of signal of problems. One of the strongest signals is there's a deficiency in brain-derived neurotrophic growth factor, or BDNF. And we know that BDNF is essential in the striatal cells for a number of intracellular processes, from transcriptional regulation, mitochondrial function, proteasomal function, axonal transport, a number of different areas. And we know from our collaborator, Dr. Gary Dunbar, that implanting MSCs that have been engineered to overproduce BDNF helps in the mouse phenotype of Huntington's disease. So our strategy is to use these mesenchymal stem cells engineered to overexpress BDNF as a rescue therapy, as a neuroprotective therapy in Huntington's disease. Dr. Nolta and members of her team and our team published a nice review about the use of MSCs as proposed therapeutic for Huntington's disease. The BDNF platform is the one that we're using. There also is a platform for small interfering RNA that we're very excited about. So we submitted a grant back in January to CIRM to develop a phase 1 trial using exactly this model in patients with Huntington's disease. We went to the ICOC meeting in July and sat through the day and listened to the family members come up and speak about the pain and suffering and fear of this disease and how badly they needed treatments. And we were there for the very emotional moment when we were told that the grant was approved for funding, and there was confetti and a lot of happiness in the room. But as Dr. Nolta and I know well, now the really hard work begins. So we are very fortunate to have a GMP facility at UC Davis, which is state of the art, if not state of the planet. It's an amazing facility, and the idea is to use it for manufacturing of cellular products. There are currently 6 stem cell trials that are using cells produced in the GMP at UC Davis, and we have batches of the MSCs and the MSCs that are engineered to overexpress BDNF baked at this facility. So the idea will be to harvest these stem cells, the mesenchymal stem cells, from bone marrow from qualified normal donors, to culture them and transduce them in the lab, to test them and store them, and eventually to have them ready for a phase 1 clinical trial. In the trial, we plan to deliver the cells through a catheter that will be delivering them into the striatum bilaterally in the caudate and putamen. Those are the 2 nuclei that make up the striatum. This will be a relatively small surgical procedure, but not without risk. The idea will be that these cells will home in on the sick and dying striatal neurons, the medium spiny neurons, and deliver BDNF and help to provide neuroprotection. So if we go back and look at what we're really doing here, this is from the CIRM website, the pathway of going from basic research into clinical trials. Dr. Nolta had a grant, received a grant back in 2009 for working on the MSCs with small interfering RNA. That's come forward, and her work is blossoming in that area. Based on the work that she's been doing in her lab, we were voted for funding for this particular trial. We have 4 years to do this. In year 1 and year 2, we will be completing the IND-enabling studies for the cells, and our goal is to have FDA approval within 2 years. One thing that's unique about our plan is that we're going to be enrolling a group of patients with early-stage Huntington's disease in an observational study starting immediately upon funding being received. We will be evaluating those patients clinically every 6 months with standard instruments of clinical measures, brain imaging measures, biomarkers, cognitive markers, so that we can establish a rate of change for each of those different areas in each patient. The idea being when we go into our phase 1 trial in 2 years, which is primarily a safety trial, we will also have some measures of potential efficacy. And because there's a great deal of variability in the course of Huntington's disease, having very robust data before implantation will enable us to both monitor for safety more carefully and also to look for signal of a clinical benefit. Future directions, as I mentioned, we plan to go forward with the MSCs engineered to produce small interfering RNAs that would be neuroprotective and perhaps more in terms of treating this disease. We're going to be learning a tremendous amount in doing this groundbreaking trial. And we're going to be learning a lot about Huntington's disease, but this has applications also for related diseases, genetic ataxias, which have similar disease mechanism. Ultimately, I think for other more common diseases like Parkinson's disease and Alzheimer's disease. We are very blessed to have a fabulous international group of collaborators working with us on this grant. We want to thank Athersys for their production of the MSCs and working very closely with Dr. Nolta. We have a collaborator in France, Dr. Anne-Catherine Bachut-Levy, who has tremendous experience in doing fetal cell transplantation studies in patients with Huntington's disease. We have a very good group of people at Mass General led by Steve Hirsch in doing biomarker measurements for us, and Dr. Elizabeth Aylward at Seattle Children's Hospital will be doing imaging work for us. And finally, Julie Stout from Monash University in New Zealand. These are the top people in the field, and they are excited to be working with us on this trial. So I want to thank the families, the patients and families more than anything for their courage and for working with us so closely and for their advocacy for stem cell research. There couldn't be a more dedicated group of people, and their work truly inspires us. And I want to thank you very much for your attention.